Okay, so before we get started for today, just want to tell you what the plan is for Thursday because that's changing a little. So I know a couple of you have emailed me about being out on Thursday, and a couple more of you have a couple of labs to make up. So what we're going to do on Thursday is I'm going to set out all of our old lab materials because you also have a practical coming up after break. So those of you who have work to make up, you can work through the labs that you've missed. And for everybody else, what I'd like you to do is to go through those old labs, make sure you understand them and begin sort of practicing for your practical. Totally cool with me if you take pictures of the materials we looked at. So anybody who's going to be out, I recommend picking a friend or a buddy and asking them to take pictures of anything that you missed or didn't understand, or just go through and kind of film the whole thing so that you have materials that you can start to work through and get ready for that practical that's going to happen after break. Sound good? Okay, cool. So that's what we'll be doing Thursday. So when we come back for the second half of the semester, we're going to be talking about this group that I'm about to introduce to you now, the angiosperms. So our angiosperms are going to be known as the division Magnoliophyta, technically. Okay, and these are going to be our flowering plants. Okay, so from before, we know that our major groups of vascular plants, which are our plants with tissue that can carry water, carry nutrients, xylem and flow. I'm sorry, I'm walking around the room looking for my clicker. There it is. Okay. We're our pteridophytes, the ferns and the fern allies, our gymnosperms, which were our division Ctenophyta, so anything that has seeds that doesn't have flowers, usually going to have cones. Now we're moving on to seed plants that have flowers. So this is our new reproductive structure. Okay. So that is our Magnoliophyta. Okay. So we're going to see that in the evolution of angiosperms, we're going to see the development of this structure, the flower, and we're going to see this structure forming around the seed, the fruit. So a cool thing about the evolution of angiosperms is that when you compare it to the evolution of other organisms, like in the fossil record, you can see an explosion of like pollinating creatures. So things like bees and wasps, beetles, locusts, and bugs, butterflies are all going to have this sort of explosion of diversity around the same time as we see the evolution of this flower. So our synapomorphies for our angiosperms, so their shared derived characteristics, so we're thinking like new mutations, are going to be something called double fertilization and formation of an endosperm. And we'll talk about that also after break as we move on to thinking about how fruit forms. So some of these terms you'll see a couple of times, so don't worry too much about too much jargon today. I am also recording, so you can come back to this after we've talked about these things a bit more. Second angiosperm synapomorphy is going to be a structure called the carpel, which is going to be related to a structure called a pistil, which is basically just a bunch of carpels fused together. We're going to see stamens with paired pollen sacs, and then we're going to see specialized companion cells in the phloem, as well as vessel elements in the xylem. It's not listed as a synapomorphy of angiosperms, though, because it was also found in Cnidum, which was one of our gymnosperms, potentially depending on how we reconstruct the phylogenetic tree. So our diversification of flowering plants is happening relatively recently. So we can see that this is our most recent group to branch off here. They're branching off from a common ancestor with gymnosperms sometime about like, guesstimate what that is, 150-ish million years ago. We're gonna watch a little video about the evolution of flowering plants, and then we're going to talk about some flower structures. When we come back after break, we're going to be actually looking at flowers as our first thing.
While browsing dinosaurs grew in size and range, plants were undergoing their own evolutionary breakthrough. This is Westwater Canyon in Utah. Ancient plant fossils of an entirely new variety have surfaced here. Tree, Laura gymnosperm. These plants had oval-shaped leaves. Their arrival would revolutionize the plant kingdom, and in time, alter the ecology of the earth. Biologist with Chicago's Field Museum of Natural History has been sifting through layers of time, studying the origins of this great change. Rocks from the Utah desert are thoroughly washed in water, then gradually dissolved with weak acid to separate stone from plant life. Soon, a tiny flower petal shaped like a tulip emerges. The engines for flowering plant. The ancestor of the modern-day magnolia may be the earliest flower on record. Inside the flower, a tiny gold bug coming out of the pupal stage. It's likely that relatives of this insect contributed to the revolution of flowering plants. Jurassic forests added a sumptuous tape, goldbug, and other new insect life. Pollen from flowering plants drifted through the forest like a soft rain. As the tiny insect moved from plant to plant, pollen stuck to its legs was delivered to the flower's reproductive pistils. You see, at the helm of the goldbug, flowers spread across the landscape. Plants that received enough pollen developed a range of distinctive colors, shapes, and scents to attract more insects. In his outdoor laboratory, Cornell University biologist Dr. Carl Nicholas observes nature's age-old alliances at work. He believes that Earth's early plants and insects formed relationships for the same reason: mutual survival.
Insect pollination allows a plant to transport pollen from one individual to the next, using the brain of the insect to identify the same kind of plant as the donor of pollen. So, for example, you can have angiosperms that are separated by many miles. Two flowering plants of the same species, separated by many miles, and the insect can transport pollen over that mileage. Plants can't move, but insects can. Gymnosperms tend to rely on wind for transport of pollen, so they can transport their pollen only a short, relatively shorter distance. So you can get widely scattered angiosperms that are still reproductively successful. If you widely scatter pine trees, they're not as reproductively successful.
The new flowering angiosperms had another advantage over other plants: the speed at which they would produce. Gymnosperms, like cedars and pines, pollinated on the wind and gained an evolutionary foothold millions of years earlier. But once their pollen reached the pistil, it took a full 6 months to a year for the fertilization process to complete. The new flowering plants were dramatically more efficient. Once the pollen grains reach the stigma of the pistil, they germinate, extending a pollen tube to deliver sperm to the ovum. Fertilization takes only 3 minutes after the initial pollen. Even the slowest of plants fertilize within 24 hours.
During the Cretaceous, for the very first time, there was a partnership that was established between flowering plants and insects. Before that time, it was an antagonistic relationship. The insects were eating the plants. But with the evolution of flowering plants, a kind of hand and glove relationship began to evolve. Flowers started changing their reproductive structures to accommodate the insects that were visiting them. And insects were beginning to change their morphology, their shape, to accommodate the visiting of flowers.
Nectar produced by flowering plants is a powerful lure for their partners in evolution. With them, new methods of pollination. Only one type of bee can help fertilize this gingerbread. Using its long tongue, it skillfully opens the petals of this flower. Pollen inside sticks to the bee's head and is later delivered to other flowers. The bee, in turn, is rewarded with nectar, off limits to other species. Through co-evolution, specialized one-on-one relationship between flowering plant and bee is established. The cooperative effort produced an explosion of insects, flora of virtually infinite texture and design. Dominating the ancient forests, insects and plants working together would survive the ages. Summer in Hokkaido, the northernmost island in Japan. Season with sweetbriar blossom blooms. The relatives of the planet's early fruit bearing. I respect to evolutionary issues. Angiosperms one species. Their first trial, attracting insects who would distribute their seed. Now, with the advent of fruit-bearing plants, came new partners in evolution. Both plants and animals benefited from this exchange. Ecologist Dr. Yumoto of Japan's Kobe University has been conducting field research in Southeast Asia and Africa to study how plants and animals interact. Yumoto has observed the symbiotic connection between flowering plants and mammals in tropical rainforests. Small food consumed by small animals like squirrels. Large fruit attracts monkeys and chimpanzees. Co-evolution to meet specific needs. As life becomes more complex, flowering plants and animals develop more exclusive relationships with one another. As large as football, this giant fruit is a treat for the modern elephant. But if elephants can feast on angiosperms, why not other large creatures like dinosaurs?
もし恐竜というのが出産をして、dinosaurs have formed a symbiosis with plants by spreading seeds and contributing to the reproductive process, then plants would certainly have made an adaptive effort to create large fruits to have their seeds dispersed more efficiently by them. But there is no evidence from existing fossils, either of plants or dinosaurs, that indicate there was ever a plant that benefited from such a relationship with dinosaurs. So, as angiosperms established an intimate relationship with mammals, or as a close interaction emerged between angiosperms and insects, their pollen agent, there's a good possibility that dinosaurs were left out of the game and lost footing in the ecological system.
Didn't really seem like the end, but we'll end there. Okay. So we are going to continue talking about the flowers of our angiosperm. Okay. So here's where we find ourselves in the tree. So when we left off, we talked about our mosses, the bryophytes, the pteridophytes, the ferns, the gymnosperms, basically pine trees and related groups. And now we're into the angiosperms. So we're going to be walking through the tree of angiosperms for a while. So we're going to be going through a bunch of different groups, starting with our basal angiosperms and progressing through. Okay. That said, the angiosperm phylogeny is a lot more complicated than that little colorful image I just made you, and it gets regularly updated. So changes pretty regularly. The most recent version, I think, I didn't check this year if they come out with a new one. As of last year, the most recent one was from 2016, which was version 4. So it's regularly updated by a group called the Angiosperm Phylogeny Group, which is a group of scientists that all works together to come up with like a consensus phylogeny for people to work from. Okay, cool. Okay, so we still have seeds. Now they're going to be covered by fruit. A fruit is going to be a mature, ripened ovary. Within that ovary, it's going to contain the seed. So from last time, hopefully we remember that a seed is our plant embryo. So that seed is going to be made up essentially of a fertilized egg cell or a fertilized ovule, which is what you see labeled in that picture of a grape. Okay. So as we move forward, we'll move from our flowers into our fruit. But before we get there, we want to talk about those flowers themselves. So angiosperms are defined as our flowering plant, which means that our flower is really going to be the reproductive structure. So our flower is going to be able to contain male parts or female parts or both. So this is kind of like that separation between monoecious and dioecious that we started looking at as we were looking at the gymnosperms at the level of a tree. But now we're having this happen within a single flower. We can either have a flower that is male, a flower that is female, or a flower that has both. When you look at pictures of flowers to label, you're usually going to see a flower that contains both male and female parts so that they can label them all out. But a flower you see in the grocery store may or may not have both male and female parts. Okay. So the female parts in the flower are going to be the ovary and ovule and related structures. The male parts are going to be descended from those microsporangia that are going to eventually become sperm. We're going to have some very specific flower words to refer to all these associated structures. Right there, we have an angiosperm life cycle, a basic one that we'll continue pulling up a couple of times to kind of walk through bits of it. Okay. So we're going to talk about the parts of the flower first. So the female parts are technically referred to as the gynoecium. Gynoecium are the female parts. The gynoecium is going to be divided into units called carpels. Okay, so the carpels are parts of the gynoecium. And a single carpel is going to be composed of a stigma. So the stigma, where's my pointer? Okay, I'll just use this. Okay, so, whoops, the stigma is going to be this part at the top of this central kind of tube that we see here. So the stigma is going to be kind of sticky. We'll talk about its role in fertilization in a little bit. Then we're going to have a style. So the style is like this stalk leading us down to our ovary. So those three things together would be a carpel. And you can see that gynecium here. So those would be the female structures in this flower. The female structures would be here in this flower. Sometimes you will see the word pistil referring to female parts of the flower. The difference between a pistil and a carpel is that sometimes we're going to have multiple carpels fused together. So here in this image, for example, our stigma is kind of cut off a little bit. You can see the edge of one here. This is a style leading down. And we can see here that we have multiple ovaries kind of fused together. Okay. So this might be considered one pistil with multiple carpels within. So female structures, stigma, style, and ovary, creating carpels, which may fuse together to form compound pistils, or you may just have one. Now we're going to add on our male parts, which is our androecium. So the androecium is going to be composed of a stamen. So we're again going to have a stalk-like structure. These are going to be thinner. So here we have a stamen. There's a filament, just a little bit, leading into a cap, where we're going to see the pollen. That cap is called the anther. Like those ones I can pull off on the flower model. And I just did pull one out. Maybe only one was blue, so it's a little floppy. Okay. So like this is our stamen. So the stick part is the filament, and then this yellow part on top is the anther. Great. So to that point, when we're thinking about whether we have male parts or female parts, we're now going to call these flowers either perfect or imperfect. So a perfect flower is going to have both the male and female parts. So you can see that here on this image. So we see a little bee sitting on the stigma, got this stalk called the style, leading us down to our ovary, which has ovules within it. Okay. So those were our female parts, or our carpel. Then we also see the male part. So we see a stamen composed of an anther, where we eventually have our pollen and that filament. So because we see both together in the same flower, this is perfect. This is the perfect situation. Okay. Our imperfect flowers are only going to have one set of structures. So in this image, we can see that we have a stigma, style, and ovary, but there's no stamen, there's no filament, there's no anther in the flower. So it's imperfect, basically because it can't pollinate itself. It can't do both parts of pollination. Has an ovary and these ovules to be fertilized, but it's not going to produce pollen. It's not going to produce these male structures. So when we think about perfect versus imperfect flowers, a cool thing that's used in biology, like to study genetics, right, is that flowers can self-pollinate. So Mendel's pea experiment relied on this property of flowers. So he was looking at perfect flowers, which can self-pollinate because they have both male and female structures within the same flower. I drew this picture, which is not me. I think drew that arrow in the wrong way. But you can see the female structures and the male structures in this self-pollination image, versus you only had imperfect flowers. You'd have to have two of them. Perfect flowers can also cross-pollinate, so you can also have genetic mixing, but they don't have to. However, when you look at a flower, the most obvious thing is actually not probably going to be the reproductive structures. So the reproductive structures, like in this model, right, are all kind of in the center, right? Those like long hairy bits, right? So those are the reproductive structures. Here we have fused carpel, female structures in the center, surrounded by these stamens, the male structures. But probably what you're going to notice is petals, right? So the petals are brightly colored, designed to attract pollinators of various types. They're very beautiful, depending on the flower we're looking at. So the petals, kind of like a modified leaf almost, fusing usually around the edges of these reproductive parts. We also are likely to have people. So if I turn this model over, see the petals are pink and colorful here. This is the same as on the image. Underneath, you can see that star-shaped structure of those like green bits coming off. Those are the sepals. So the flower is also likely to have these usually green parts kind of surrounding the petal. Now we will have words for describing all the petals together or all the sepals together. So all the petals together are going to be called the corolla. All the sepals together are going to be called the calyx. And if we're trying to refer to just like all these sort of leaf-like structures around the reproductive parts, we're going to refer to that as the perianth. So you can think peri like next to anth, think about like the anthers or something. Okay. Couple more terms. So below where we have our reproductive tissue, we may also see a structure that we call the receptacle. So we can think of this as kind of like an attachment point for all these flower parts. Flowers usually have like their own little stalk attaching them to the stem. So that's called the peduncle or sometimes the pedicel. Okay. And sometimes we're going to see a kind of tube form. So sometimes this is referred to as the flower tube. The technical term is the hypanthium. So in this image, you can see this green kind of bowl structure. So we see a green sepal here, we see a petal here, leading us into this tube. And this is the hypanthium. And we can see that on the real flower as well. So we see a petal, we see our stamen, and we see a sepal, all of these kind of attaching and leading us into this tube, that hypanthium. And then at the center, we see our ovary, our style, and our stigma. So these are some other parts that we want to know the names of as we start to talk about like where is the ovary relative to the rest of the parts of the flower, where are the sepals, where are the petals, how are they divided, which are things we're going to have to look at as we start to identify the flowers of specific plants in their specific groups. Okay. So to that end, we are going to be sometimes describing how the ovary is positioned relative to these parts. Okay, so we're going to see these words hypogynous, perigynous, and epigynous. And these are all referring essentially to how the ovary is positioned relative to the other parts and mostly to that flower tube structure, that hypanthium. So on this image, we can see the ovary in this beige or light yellow kind of color. So the ovary is the swelling part, the style is the soft part, the stigma is the little bulb on top. And we can see that in each of these. So ovary, style, stigma, ovary kind of fused in here, style, stigma. So when we look at these things separately, I'll pull them out for you. So in the hypogynous flower, so hypo usually means low or less. So we're thinking below. Specifically, what we're talking about being below is all the other parts of the flower. So we can see here in this image, our ovary is superior to all the others. And we will see sometimes flowers described as a superior ovary, an inferior ovary, things like that. So that's why I use that wording here. So here we see this ovary sitting superior to the attachment point of our stamen, petal, or sepal. This is the most common thing that you're going to see in flowers. So if you're looking to guess hypogynous. Okay. So we can see that structure here in the real flower as well. So here we see the ovary a little narrower than in some of our cartoons, leading to style stigma. We can see those other structures attaching inferior or below that ovary. So that's hypogynous. Next, I'm going to jump to the part C of that first image, because C is kind of an in-between. So our epigynous flowers are going to have structures attaching kind of at the top of the ovary. So the ovary here would be considered inferior to the attachment of all these other. So here we can see the petal coming in, the stamen coming in, the sepals coming in, all here, kind of at the level of the middle of the style. And then we can see that the ovary is basically contained within this structure, okay, this lower structure. So here we see what that would look like potentially in a real flower. Here we see an inferior ovary. Here we see all these other structures inserting above the ovary. So that's an epigynous flower. Then our third part is kind of an in-between, more difficult to identify, is a perigynous flower. Okay. So here we see the ovary kind of contained within the hypanthium, so this tube or kind of cup or bowl structure. So a clear image of this in a real flower is here. So the hypanthium here, where you have those sepals fusing and forming this little tube. You can see that the ovary is sitting in it and leading it up. So that's what it looks like when we have a perigynous flower. Okay, so we're going to have these structures starting roughly at the same height as like the center of the pistil, or in general, these female structures. I pulled on a rose here because I find the rose particularly confusing. It is technically defined as a perigynous structure. The reason I find it a little confusing is you have to really pull apart that hypanthium from the ovary. You can see it's kind of fused in a little. So it does look a lot like the epigynous structure, but technically a rose is perigynous. Cool. Other things we can look at or describe is the symmetry of flowers. So flowers can be described as actinomorphic, which is radial symmetry, zygomorphic, which is going to be bilateral symmetry, or just asymmetric, you can't draw any lines. Okay. So when we're talking about these types of symmetry, we're talking about you draw a line through the flower, is it the same on both sides? Okay. So for our actinomorphic flowers, they have radial symmetry. This is the more common condition for a flower. What we're saying is you can draw a line, splitting it in half and have it be the same on both sides, but you don't have to draw that line in any specific spot, right? You can draw it here, the same on both sides. You draw it here, still the same on both sides. Here, still the same on both sides. So that's what we mean by radial symmetry. Those lines where you place them doesn't really matter because the symmetry is kind of radiating out. So that's actinomorphic. For our zygomorphic flowers, you're going to have to draw that line in a specific spot. So zygomorphic flower will have two sides that are the same, just like mirror images of each other. But you have to be careful about where you draw the line, or you don't end up with this symmetry. Sometimes we'll see that there are maybe two spots where you can draw a line, but nowhere else. But our best examples of the zygomorphic flowers is one line down the center would divide them in half. So our asymmetric ones are just going to be weird, so I don't have that pulled out separately. So you can see that there's not really any line here that would divide this, giving the mirror images. So the flower is part of our life cycle of the angiosperm. Okay. So here we have our flower. We know that this is, because it's part of the reproductive cycle, going to lead us eventually into our gametes, both female and male. So the gynoecium and the female part, ending in the ovary and the ovule, are eventually going to lead us into an egg that can be fertilized. The fertilization in the spectrum is down here. So all this female process is on the left side of the image. The male process begins with the anther and someone on top of it. And that's going to lead us down through this male pathway, eventually creating sperm. which are going to land on top of the stigma, which is sticky on top of this part of the gynomecium. I'm going to extend down to fertilize the ovule. So in this example, we have a perfect flower. We see both male and female parts, but depending on the species, this might be imperfect flower. You might need two different flowers, a male and a female one, to have this whole process. So you can see that we still have this sort of alternation of generations kind of thing going on. So we have like analogous to our sporophyte and gametophyte generations, and we have a level where we have meiosis. So we have that division to get to that haploid condition with our egg and our sperm. Okay. So we're going to talk first about the female process, and we will review fertilization as we move into fruit, because that's going to create the different parts of our fruit. Okay. But when we're talking about the female half of this life cycle, so we're thinking about the gynoecium, we're thinking about the stigma, the style, the ovary containing the ovules. Okay. So we're starting in the ovule, and we're going to start with a megaspore mother cell. So this megasporangium, referring to a diploid cell, so that's 2N, right? So it's diploid, just like our sporophyte generation were. So this diploid megaspore mother cell is then going to undergo meiosis. So we're going to undergo 2 cycles of division, taking us from diploid to haploid. So we're going to go from diploid to haploid as we go through meiosis, creating megaspores. Weird thing that happens is that while we create 4 megaspores through meiosis, we divide 1 cell twice to get 4 cells. 3 of them are going to get aborted, and we're just going to have 1 surviving megaspore. That surviving megaspore is going to undergo mitosis until there are 8 cells. So those 8 cells are going to be considered 8 nuclei. They are then going to become different structures within our eventual fruit. Okay. So of those structures, we're going to form some polar nuclei. So these polar nuclei are eventually going to become something called our endosperm. I'll talk about that as we talk about our fruit. We're going to have an egg. That's the part that's going to get fertilized. Okay. So that's going to form a diploid zygote when it gets fertilized. So diploid, because when we get that information from our other gamete, the sperm, that brings the other half of the genetics in. Diploid. Zygote, because that's just the word for the structure that we have after fertilization. But we're also going to have some other structures. So we're going to have some structures called synergids that are going to essentially give a signal to the pollen about where to go. So those are here at the base that we can see. We're also going to have some antipodal cells. So we can see those up at the top, and those are going to nourish the growing egg cells. There could be meiosis first, then mitosis, all of this happening before we get to our fertilization. On the male side, in the androecium, we're looking at pollen, essentially. So our pollen is a microspore, essentially. It's going to originate from mother cells in the pollen sac. The pollen sac is contained within the anther. So here we can say we like slice the top off one of the anthers. Right, so we like slice this here, right? Essentially, we made a cross section through this anther. Okay. So we're now looking at pollen sacs in the top here. And we can also see that on the slide up here. So this like histological slide is a lily arch. So we slice open the anther of a lily. So we can see that we have these pollen grains forming within the anther. So on the male side, we also have meiosis. So we're creating 4 haploid cells, a structure called a tetrad. Don't worry about that part. Okay. The word tetrad. But that's the technical term. And then through mitosis, we're turning these into individual pollen grains. A pollen grain here is actually not really just one cell. So as we form our pollen, we're going to end up with one cell that's the generative cell and one cell that's the tube cell. And then the generative cell is going to form two sperm. So we're having a tube cell as well as generative cells that become sperm. So that's important because when we get to the face of this image, right, we can see these sperm cells, but we can see that in this pollen, we also need to form this pollen tube that is going to project down the style of the gynoecium to get to the end. So what we see happening here at the base, we have a stigma up here that is sticky and sugary. This is part of what attracts those pollinators, right? They're trying to get to the sugar sitting on top of the stigma. They got a little pollen all over their feet, maybe. When they sit to get this sugar, they deposit these pollen grains. So that pollen is going to penetrate into the style of that pollen tube, descending down through that stigma, through the style, to get down to the egg. And angiosperms are going to undergo something called double fertilization. So one of those sperm is going to get to an egg, become a diploid zygote, eventually forming an embryo. But the other sperm is going to unite with two of the other cells that we had in the gynoecium forming that were not the egg. So the other sperm is going to unite with the two polar nuclei. And those polar nuclei are going to join together with that sperm to form something called the endosperm, which is going to provide nutrients to this growing embryo as we fertilize the egg. I'm going to look at all of this again as we look at the fruit. But zoomed in here, we have that process. So now we're looking stigma, style, and ovary. So these central parts of the flower, these female parts, the gynoecium. So we can see the pollen grain landing on top of the stigma. So this is sticking here. We have a generative cell that's going to form our sperm, and we have a tube cell. Okay. So we can now see that the tube cell is forming this pollen tube that's descending down the style to get to the egg. Okay. We can see the sperm traveling down that pollen tube. Okay. So the generative cell traveling down that pollen tube and forming its two sperms as it goes. When the sperm get all the way down into the ovary, we're going to have that sperm, one of them, joining up with an egg cell, fertilizing it. The other sperm cell doing double fertilization, fertilizing those polar nuclei. Only one of the things becomes a zygote, which is that joining of the egg and the first sperm. Okay, so here we see the forming embryo cell. Okay, so we can see fertilized egg eventually going to become our seed, essentially up here. We can see those polar nuclei, sperms that come in. This is where we're going to form our endosperm, which is going to become a food source. So that's what we got for today. So we will be looking at a bunch of these structures when we come back. And on Thursday, we're going over all the old lab material. Also, if I could say, if anybody's going to be absent, to ask someone to take pictures of lab stuff. If you guys want to send out an email thread and split that up as a class, that's just a suggestion of something you can do. I'm not going to arrange it for you, but I don't mind if you do it. Don't believe in duplication of work.